Hello everybody, in today's video we're going to go over the basics of genetic engineering, and this is the kind of stuff that's been going on since the 1970s until today. And the very simplest version of this is to genetically engineer bacteria. And so what we're going to be discussing is the kind of things that led to the birth of the modern biotech industry. So here we have a basic bacterium. Don't forget, we've got a single circular chromosome. More importantly for today, there are additionally little loops of DNA called plasmids. Now, evolutionarily, these are really cool if you think about it, because bacteria being asexually reproducing organisms, the plasmids actually give them the ability to perhaps transfer small amounts of genetic material between related and even unrelated strains of bacteria. So these additional little loops are going to be what we're going to be using today. Now, to give as a framework, we're going to imagine that we're going to try to mass produce specific human proteins for therapeutic purposes, right? We're going to sell these to people who have particular disorders that they're struggling with. And some examples of this that have been profitable, I suppose, are things like insulin, growth hormone. We've got erythropoietin, which is used to trigger red blood cell production in people who have issues with that. And if you have hemophilia, clotting factors, specific varieties may be useful. So all of these have been marketed and sold and have been very profitable for the companies that have produced them. So what we're going to be doing is working our way through the whole process, and it takes about 6 steps. Obviously, we're leaving out a lot of details, but the first step of all is to get your gene, and you need lots of it to mess with. So we're going to copy that. The process that you've learned about already is called the polymerase chain reaction, and very simply, into a tube, you put your gene of choice, wherever you're going to get it from, along with the raw materials for making more DNA, so deoxynucleotide triphosphates. We've got to have primers to specify where you're going to do your copying. And then lastly, a heat-stable polymerase. You put these into the tube, and assuming that you have good template DNA, and I'm not going to go into the details of this, but you better have a version of the gene that's called cDNA, which is the version of a eukaryotic gene that doesn't have introns in it. But if you do so, you just take this tube, shake it up, put it into a thermal cycler, which will go through the necessary temperature cycle to allow for copying of this gene. So at this point, you have billions of copies of the gene. That's step 1. Step 2, though, is to have something to put that gene into, and that's your plasmid. So you're going to start with a plasmid that already has a gene in it, and we're going to cut out whatever's already there. To cut the DNA of this plasmid, we're going to use restriction enzymes, which you've learned about previously. So if this is our basic plasmid to start with, the green arrow represents a promoter, and whatever gene's in it is the pink bar there. And keep in mind, there are plasmids for expressing lots of proteins that are already out there. So if you started with a plasmid for expressing growth hormone, you could just cut out the growth hormone gene and pop in an EPO gene, or pop out the EPO gene and pop in a clotting factor gene. This is not the most difficult thing to do, given modern techniques. So let's say we've cut that plasmid. The issue at this point, though, is we've got the plasmid linearized, but we also have those little gene bits, the gene that we cut out that we're trying to get rid of. So now we have 2 different fragments of different sizes. So the one we want in this case is the longer of the 2 fragments. Our technique for isolating the particular fragment that we want is called gel electrophoresis. As we've discussed, this is what's going to help us separate fragments by size. The larger size move slower through a thick gel, while the smaller ones will be able to maneuver their way through much more quickly and efficiently. Keep in mind that this is negatively charged DNA, and a current towards a positive electrode is going to help move those DNA fragments along. All right, so now we've got copies of the gene and lots of copies of the cut linearized plasmid. If you have them and throw them into the same tube, they will unfortunately not magically stick together. not to any significant degree. So we're going to have to throw them together with an enzyme that can put those ends together covalently. You've already learned about this. So this is going to be ligase. So we're going to ligate these fragments together using the appropriate enzyme that you learned about when we learned about DNA replication. Remember, this is the enzyme that connects the fragments together when we're doing DNA replication. All right, so we take these fragments, place them in a tube, add in this enzyme. The most effective version of this enzyme actually comes from a virus that we use currently. But hopefully at the end of this process, a few hours later, we've at least got some percentage of the fragments that have been ligated together. Okay, next, it seems like we're almost there. At this point, we have to actually get this plasmid into the bacteria or whatever organism we're trying to get it into. So the process of putting DNA from one source into another is called transformation or transfection. Transformation is the term we use with bacteria. Transfection is more commonly used when we're talking about eukaryotic cells. All right. Now, to do this, you really have to mess with your target cells. This is not a gentle process. We basically screw with the membranes of the bacteria. And we can do so using high temperatures, crazy salt concentrations, or even effectively electrocution. So we're going to electroporate or heat shock are the terms that are typically used. Regardless, we're messing with their membranes, and at some incredibly low percentage, maybe 1 in a million of the bacterial cells, if you're lucky, by chance, gets one of these plasmids diffusing through their messed up membranes into their cytosols. So that's the hope. So if you get, again, 1 in a million, that's not bad transformation likelihoods. So you get to take all these bacteria. Some have been transformed. Some of them have the plasmid. Some of them do not. And you'd imagine just spreading them on a petri dish and growing them up. Here's where things get, well, not difficult, actually pretty easy, but difficult to imagine at first. You've got all these different colonies of bacteria growing up on your dish. You know, maybe there's 100 or 200 or 500 little bacterial colonies, each with a billion bacteria in it. The issue is we don't know which of these dots, these colonies of bacteria, actually got the plasmid. So this brings us back 50 years. And 50 years ago, when Cohn and Boyer were developing the first recombinant plasmids, they did something really clever. And we've been using the same trick ever since. There's another gene on the plasmid. And this was actually the only gene on the original plasmids that Cohn and Boyer used. So this gene in orange, you can now see on the original plasmid and on all the fragments since, is the essential gene for this next step. This is an antibiotic resistance gene. It is a gene that, when expressed, produces a protein that gives the bacteria the ability to survive a specific antibiotic. Whether it's ampicillin or kanamycin or spectinomycin, whatever it is, this is a gene that protects those bacteria. The key here, though, is if you've got the plasmid, then you're safe. If you didn't get the plasmid, you are in danger, especially if you spread that antibiotic all over the plate. What you're going to do at this point is you are effectively going to annihilate any of the bacteria that didn't get the plasmid at all. Anything that doesn't have the plasmid will not survive the antibiotic, will be killed off, and the only things that actually end up growing on the plate, there's not much on the plate, really, are the ones that actually have the plasmid. And just as importantly for us, these are the ones that can now make the protein that you wanted them to make. So that pink gene, whatever it is you wanted to put on there, the blue gene, whatever it is you wanted to put in there, they are hopefully making that protein now. So this is the end success. So what you can now do is just pick that bacterium off that plate, grow massive vats of it, give them the chemical induction that they need to know to produce the protein, then take a few 100 or 1,000 liters of these bacteria, blow them up, purify the protein, put it into little bottles, and sell it for hundreds of dollars each. All right. That's the whole process, folks. Hopefully that was helpful to you.